I'll do.
Yes, so if you don't mind just clicking through slides, and I'll let you know when to move on to the next slide.
Yes.
And then we'll go through it that way. Sorry for the little bit of the delay today.
All right, everybody. Welcome to the third session of Nutrition, Wellness, in the Brain. Again, sorry for all the tech issues this morning, or I guess now this afternoon. This series is a collaboration between U of I Extension and the Interdisciplinary Health Sciences Institute at U of I. Really, this all worked just beautifully last week, so they're with us. I don't think every week will be like this. We do appreciate any feedback that you're willing to give. We will put in some surveys in the chat box. If you're new to the series and this is your first time, there'll be a survey for first timers. And then at the end of the session, there's a survey for everybody to take to learn more about what people want to learn about in regards to brain health, as well as if they are looking for more information on the topic that we presented on today. I do want to do a quick little commercial. We do also have a Discover Wellness series going on. And for people who are working from home, or for anybody out in the community. So I'm going to pop that in the chat box. And if you want to, you can check out that educational webinar series as well. And then without further ado, I'd like to introduce our speaker for this series, Corinne Cannavale. Corinne received her Bachelor's of Science degree in Molecular and Cell Biology with a minor in chemistry from U of I. She's currently a PhD student in the Body Composition Nutritional Neuroscience Lab. And her primary research investigates how memory can be enhanced through diet and exercise. Today, her focus is going to be on childhood nutrition. So I'm going to pass it over to you, Corinne. Take it away.
Great. And just a disclaimer, I do have a dog, and I'm at my apartment now. So if you guys hear a dog squeaking on toys, because he likes to use his loudest toys while I'm on Zoom, that is what you're hearing. So Chelsea, you want to go ahead to the next slide? There are some animations, so just that'll also be something to keep in mind. So I'm first going to talk briefly about cognitive function and how we assess cognitive function during or while studying nutrition. So I'm going to talk about three kind of domains of cognition that we like to study in our lab and also some labs that we collaborate with. So the first one being memory. So this is kind of the idea of how well our brain can hold on to information. And so there are two types of memory that we study a lot. And so these are short-term and long-term memory. And the thought is that these are controlled by two different brain regions, which is why we study memory in two different ways when we are assessing these things in nutrition. And a good example of how memory might be used in childhood, especially in like an academic setting, would be after reading like a book or an article, kind of having to recall that information to write a book report or do a presentation. We also study something called mental flexibility or cognitive flexibility. And basically this is just how well our brain can accomplish multiple tasks or switch between two different tasks. So kind of the idea of multitasking and our ability to switch between two different rule sets. So when we study this in the lab, it is a lot more theoretical. And so we're doing some computer games that involve switching between two different rule sets while pressing some buttons and playing those games. But in the real world, you could think of this more as switching between two different rule sets when doing a series of math problems. So if you're switching between subtraction and multiplication and division, having to switch quickly between those in timed math sets would be a good explanation for that. And then the last one we study in the lab is attention. So how well can our brain ignore irrelevant information and focus on what's most important and what's there? And so this is really relevant for kids trying to pay attention in classes or kind of pay attention to certain tasks that they're doing. So making sure that they're really attending and focusing on information that is the most relevant for them at that time. So we have a variety of ways that we can study these things in the lab. So there are computer tests that we can do, and we have a few different imaging techniques we can do with MRI scanners and with EEG caps. But mainly I just wanted to kind of overview these domains that we study in the lab and how they relate to real life examples of usage for kids. So if you want to go to the next slide, Chelsea. Perfect. So I also just wanted to go back to a slide that I showed at the first week, if you guys were there in Logic when we were meeting in person. And this is the idea of correlation versus causation. So a lot of nutrition research is first done at a correlational level, within the idea that that can be built upon for doing more large-scale intervention works. And so a correlation does not necessarily mean that there is a causational relationship between two things. So here I pulled an example from this website called Spurious Correlations. And so with this website, basically what you can do is you can put in different data sets, and they will tell you whether or not these are correlated with one another. So you can see here that cheese consumption per capita is correlated with women who are awarded PhDs. And so you could construe this and say that as more cheese is being consumed, more women are going to be at the level to receive a PhD. But this is obviously not a causational relationship. These just happen to be related to one another. So I'm first going to talk about some correlational research. This isn't necessarily to say that the correlational relationships that we've seen are completely made up, but it is something that nutrition research then builds upon to create larger scale intervention studies where we can actually assess causational relationships. And if you want to move to the next slide, Chelsea, sorry, I hit pause. And then the last thing I wanted to talk about was just how we assess nutrition and diet in our research. So on your own, you can kind of assess, there's actually one more animation, Chelsea, sorry. You can assess your diet on your own through a lot of applications, but with research, we have a lot more sophisticated methods for doing this. So we have two primary ways that we do this, through biomarkers and self-report. So a biomarker would be something that we can biologically sample from a participant while they come through the lab. This can be through like a blood, serum, or a plasma marker. And then in our lab specifically, we do two really cool methods. The first one is assessing macular pigmentation. So using a blue light and how our eyes absorb blue light, we can assess how much of a specific nutrient is in the macula of the eye. And I'll talk about that a little bit more on a slide later on. And then we can also assess skin carotenoids using just the red light that flashes on participants' fingers. And so basically we can assess how much of certain nutrients found in green leafy vegetables are depositing in the skin with a non-invasive measure. We also do self-report measures for nutrition assessments. So the first one would be a 7-day diet record that I have listed here. And so basically participants will complete a diet record for everything they've eaten and drank for 7 days. And then we can use this really sophisticated software called the Nutrition Data System for Research. And that will actually allow us to quantify specific nutrients that they're consuming in their diet over a week. And then we can kind of generalize that to what their normal diet patterns would be. We can also do diet history recalls. These can be done farther back for 6 months or a year. These aren't recording specific things that everything was eaten every day. It's more just a frequency of how often these nutrients were consumed. And so these are also a really useful tool and are the gold standard for assessing self-report measures of diet. But you can see that there may be some problems with if you were asked to evaluate your diet a whole year prior. It might be difficult to remember how often or how frequently you ate certain foods. So they are limiting methods, but they are really useful for us in assessing diet and diet quality in our research. So we can move on to the next slide. And so just briefly, I'm going to be talking about a series of nutrients that are that have been researched in relation to cognitive function, and these are all going to be correlational studies that I'm going to be talking about. I'll talk about carotenoids, omega-3 fatty acids, and fiber, and how those are shown to positively relate to cognitive function. And then I'm also going to talk about saturated fats and added sugars and how those are shown to be negatively related to cognitive function. We can go to the next slide. Okay, so we'll first talk about carotenoids. So carotenoids are plant pigments, which our bodies can't produce on their own. So some carotenoids can help us produce vitamin A, and then others don't produce vitamin A, but they do have antioxidant properties in our body in places like the eyes and and they also deposit in regions like the brain. I wanted to briefly highlight the idea that carotenoids can't be produced in our own body. So a really great example of this is flamingos. So you can see a baby flamingo is actually more of like a gray-white color when they're babies. But because of the diet of flamingos being largely comprised of shrimp and the high beta carotene content in shrimp, that actually will cause their skin and feathers to pigment this pink color. So it's not something that they're producing on their own to get that color. It's actually something that's being achieved through their dietary intake of carotenoids. And as I mentioned, carotenoids deposit in brain tissue. So I included a chart from a paper here, which was able to quantify carotenoid concentrations in different brain tissues of deceased infants. So unfortunately, these infants had passed away due to natural causes, and the parents donated their bodies to science, and we were able to quantify these carotenoids in their brains and kind of see how certain carotenoids are depositing at higher levels. So specifically, this carotenoid lutein deposits at a really high level in the brain, larger than any of the other carotenoids that were studied in this paper. So if we go to the next slide, I'll talk briefly about macular pigmentation. So as I mentioned, carotenoids can deposit in the eye, and specifically lutein and another version of lutein called zeaxanthin deposit in the macula of the eye, and they create macular pigmentation. So in the top left, you can see a diagram of what the macula of the eye is, and actually you can see yellow pigmentation in the macula because of the lutein that's depositing there. And so this helps a lot with blue light absorption. So in the figure below, I included a figure that talks about how much blue light can be absorbed based on how much macular pigment that you have. And so basically we can take advantage of the idea of blue light absorption in the eye relating to these macular pigments. And we can assess how much macular pigment there is with a non-invasive method. So it's just an eye test that participants look into a scope, see a flickering blue light, and tell us when it stops and starts flickering. And with that, we can assess how much lutein is in the eye. So it's a really cool non-invasive method that we can use in the lab to get an actual biomarker rather than just looking at diet records that the participants are filling out. And so studies in our lab and labs that study these kind of nutrients have found that carotenoids in the eye are related to better memory functioning, faster processing speeds, improved executive functioning. So kind of the whole package when looking at memory, mental flexibility, and attention. And then they're also related to better academic achievement. And so again, these are relationships, not causal effects, but they are things that are then looking into more. So a few intervention studies in adults have seen improvements in these after carotenoid consumption. And our lab is actually doing a intervention study in child age groups right now to better understand these relationships. So the next thing I think I'm going to talk about is omega-3 fatty acids. So these are healthy fats that are found in foods like fish, eggs, and oil. And I forgot to mention, if you got the handout from Chelsea yesterday in the email, I actually included a list of some foods that all of these nutrients can be found in. And these are building blocks for the insulation of neurons in the brain. So I talked about this a little last week. But basically, there is something called myelin, and myelin is a fatty tissue that kind of wraps around our neurons and insulates them, so that signals can be processed faster down a neuron. So if you have better insulation, then these kind of electrical signals that get sent from where this green portion of the neuron is can send down faster to the bottom end to then transmit to other neurons. So we want better insulation, and omega-3 fatty acids can kind of help us on the road to doing that. Do you guys have questions about this portion? I'll give you guys a few seconds to put anything in the chat, and then I'll have Chelsea move on to the next slide. Okay. There's also, as I mentioned on the earlier slide where we talked about different nutrients, so there's also saturated fats. So saturated fats are primarily solid fats, and these are found in things like butter, that should say red and preserved meats, and lard. And these can impact how like fluid cell membranes and the insulation of neurons can move properly. So I included this really great figure from Khan Academy where they talk about the differences between saturated and unsaturated fats. So you can see that saturated fats can stack really nicely because of how straight their little like fatty chains on the end are. So they stack together really neatly, and that's what causes a more solid fat when we're looking at foods. Unsaturated fatty acids have double bonds in these little fatty tails, and so they can't stack as neatly, and that's why you get more liquid fats like oil. And when you're looking at this in relation to a cell membrane rather than foods that we're going to be consuming, you can think that it would actually be more beneficial to have these more fluid membranes, so that way our cells can move around easier and let nutrients in and out of the cells more readily. And so it's important that we're keeping this in mind as we look at the different foods that we're eating, because it can affect the way that our cells are depositing these fats into them. And so then when we're looking at cognitive function, so if you go to the next slide, Chelsea, we've actually seen relationships with memory function and fat consumption. So here I included a figure from a paper that came out of a lab at the University of Illinois, which assessed different types of fats that were consumed in diet and how that related to memory function. So when we're looking at omega-3 fatty acids on the left, we see a positive relationship between these fats and memory function. So kids that were consuming more of these fats were performing better on a memory task. On the other hand, on the right side, we see saturated fats. So with saturated fats, they have decreased memory function with higher fat consumption. So we're seeing the opposite relationship for these more solid, unhealthy fats in comparison to the omega-3 fatty acids. Do you guys have questions about this? Okay, we can move on to the next slide. So the last two nutrients I wanted to talk about were fiber and sugar. So I'll first talk about fibers. So fibers are carbohydrates that can't be broken down by the body, and they're really useful for helping our bodies regulate blood sugar as well as keeping us full longer. And so when looking at cognitive function, there have been a few studies looking at these, specifically one actually done by Kelsey and the other actually done by my advisor who I work with. And so these studies have found that fiber is associated to better attentional and cognitive control abilities. So looking at attention and children's abilities to focus on one specific item while ignoring irrelevant things. And then it's also been shown to be related to better creative thinking skills. So I didn't talk about creative thinking skills on our cognitive function slide, but this is kind of another way that we assess how a specific region of the brain that functions with memory also can affect creative thinking. And so we have seen higher fiber consumption relating to better creative thinking with that. And then when we're looking at added sugars, so on the next slide, we see kind of a negative relationship here. So added sugars are prevalent in a variety of processed foods, and they may also contain naturally occurring sugars. So I just wanted to talk briefly about the idea of what an added sugar is. So basically added sugars are sugars that are being added to a food product on top of any kind of naturally occurring sugars. So an added sugar can still be a naturally occurring sugar. It's just the idea that it's being added into something. It's not like necessarily an artificial sugar. So corn syrup is a really commonly used added sugar. So it's created from the starches that are found in corn, and then that's added to a lot of processed foods. So it still is a naturally occurring sugar. It's just added on top of any kind of naturally occurring sugars. So we get a lot of naturally occurring sugars in things like fruits and yogurts, but then there are also added sugars that can be added to things like yogurt. So so there is kind of a discrepancy between what those two things are. And I can talk a little bit more about that if you guys have any questions on what constitutes an added sugar. But again, in the study done by Kelsey on campus, we saw that added sugars were negatively related to creative thinking abilities. So poor creative thinking in relation to higher added sugar consumption. You guys have questions about any of these correlational studies before I move on? Okay. And so I now just wanted to talk about this hierarchy that we talked about at the first session that you guys came to. And so this is basically just looking at the hierarchy of what kind of evidence is the most reliable when looking at nutrition research. So you can see that correlational analyses, which are at the bottom, are more like a snapshot of our participants. So they're looking at one specific time point where we're assessing their diet at that week and their cognitive function at that week. There is then the idea of a randomized control trial. So this is looking at a more long-term effect. And so this would be more like a movie or taking a video of our participants. And I actually have one more animation, Chelsea. There you go. And so basically we're saying if we give these participants a certain food item or a certain supplement, how does that affect their cognition after that consumption? So we'll usually assess cognition prior to and after they consume each thing, so we can see whether or not there was a change. And then if we go to the next slide, I'll talk about water and kind of the essential nutrient of water and how that can then affect cognitive functioning. And I'll display some evidence from a randomized control trial that's been done at the University of Illinois. So this is an essential nutrient needed for normal body functioning, and it's estimated that approximately 50% of US children are not properly hydrated. So this is actually really problematic because water is really important for regular body functioning with metabolism, but then also is really important for proper brain functioning. So if we go to the next slide, there was a water intake study done at the University of Illinois, and basically the children that were recruited for this study went through 3 different interventions. So the first portion, they just did their normal water intake. They were basically just consuming what they would normally do throughout the day, and they came in prior to this intervention period. That way they could, we could assess changes. So after the normal intake, then the participants did both low and high intake, where they for 4 days consumed either 2.5 liters a day for the high intake, and then just half a liter a day for the low intake. So you can see they did all 3 versions of this study, and then we looked at changes for each one of these different intake periods, so we can assess how dehydration and kind of like the top level of hydration can affect their cognitive functioning. And so the study found that they saw improvements in this mental flexibility. So basically the child's ability to switch between different rule sets and do different tasks at the same time. So this, as I mentioned, was kind of similar to doing different types of math problems and quickly switching between the different rules that they would have to do those math problems. And so we're seeing improvements in this. And so that for this case, we can actually definitively say that water does improve mental flexibility, because it was more of a change that was seen after an intervention period rather than just a correlational relationship. And so then I briefly also just wanted to talk about breakfast consumption, just because I feel like we get that question a lot as people who study nutrition and the brain, and how that relates to things like eating breakfast. So on the next slide, I just talked a little bit about where the idea of breakfast being the most important meal of the day came from. So this was actually a marketing technique made by the Kellogg's company to get people to buy their cereals. So this was something that kind of was brought up. The whole idea that breakfast is the most important meal of the day was originally started as an advertising campaign to get more people to buy cereals and consume cereals. But when we kind of look at the evidence, there is some evidence indicating that this may actually be beneficial. So on the next slide, I included 2 studies that kind of talked about breakfast and brain health. So looking at more like epidemiological large-scale studies where they're just assessing children who consume breakfast before coming to school and then seeing how well they perform, we see that children who are consuming breakfast can perform better on academic skills at school. And then I also highlighted one intervention study, which I included a figure from below. And basically they had participants consuming specific regimented breakfasts that they were controlling, and then looked at their working memory capacity and executive functions after those different breakfast consumptions. And so they found that kids who were consuming breakfast at all different grade levels, I guess specifically at these higher grade levels, performed better if they were having breakfast versus if they were not. And so the studies also have kind of highlighted that certain breakfasts were better than others. So in the intervention study, they found that breakfasts that were higher in like just fruits and vegetables did not perform as well as those with like higher levels of whole grains and fibers and fats. So more balanced meals rather than just a fruit. And then in the large scale epidemiological work, where they're just looking at a large amount of kids in a school, they found that whole grains were preferred to those that were high in sugars for breakfast, with performing better on academic performance. And so just kind of going back to everything that we've talked about, these are the key takeaways for today. So the first one is just looking at these nutrition associations. So healthy brain functioning in childhood is related to higher consumption of things like carotenoids, omega-3 fatty acids, and fiber. But poorer brain functioning is related to higher consumption of saturated fats and added sugar. When we're looking at water, we see that increased water consumption and being hydrated is going to lead to better mental flexibility. And with breakfast, again, as I mentioned, breakfast being the most important meal of the day was started as food advertising, but there is some evidence that shows that breakfast may benefit a child's academic abilities. So it is something to keep in mind to just make sure that we're evaluating all of the evidence when trying to understand what could be beneficial for our kids. And then I think I have one more slide, just with my references, and then just a question slide after this. So feel free to put some questions in the chat, and we can definitely go back to certain slides if you want more clarification or just any additional questions that you guys can have. And then Chelsea put the survey for feedback today in the chat. So these are really useful just for us building sessions moving forward, and then also just for my uses with kind of evaluating what I'm going to be including in future sessions.
We did have somebody raise their hand, so if they want to go ahead and unmute yourself and ask your question, or you can type it in the chat box.
Yeah, so it looks like Divya just asked a question about water. So the study was actually only looking at water consumption. So I think the children were asked to limit their consumption of sugar sweetened beverages. And so interestingly, they actually didn't see much of a change in hydration from the low condition to the normal intake. So it's thought that maybe if they had allowed that consumption of sugar sweetened beverages, that maybe that would change. But they were asked not to consume those.
Hi, this is Ophelia. I have a question. I know we're talking about childhood nutrition, and in regards to the carotenoids, is there a
Mhm.
difference in age? And as we get older, does our body not accept or absorb carotenoids as much as in our youth?
Yeah, that's a really good question. So I included a figure from a paper which looked at infant brains. They've actually assessed carotenoids in the brains of, they're called centenarians, so people that were over 100 who were involved in a study in Georgia. And so they still do see the same trends in deposition of these carotenoids in the brain. Um, I know that it is more difficult for these carotenoids to be to be absorbed later in life, but they don't know if that's because of a bioavailability or if it's because they're having a harder time reaching the brain just with other things going on throughout the body. So I'm not sure exactly whether or not it is due to aging or due to other things going on in the body.
Other questions?
It also looks like that Chelsea had posted the previous lessons for you guys to access if you guys wanted to go back and look at those. I actually haven't had a chance to re-record the first session, but we're also going to re-record the first session so that you guys have access to that since that was done in person at Logic.
Could you develop more on how the assay to determine the level of carotenoids in our skin is about, or how does it go about?
Yeah, so it is done with a tool, we call it the veggie meter. And so basically it uses a technique called reflection spectroscopy. And so it basically sends light of a certain wavelength into the skin, and then how that scatters back to the device is kind of how it's quantifying the amount of carotenoids in the skin. So basically it's all based on pigmentation and light absorption because these nutrients have specific properties in light absorption.
And these nutrients, where do they get deposited in the cells of our skin? Like, is it like in between the cells, or are the carotenoids inside our cells, or
That's
like?
a good question. I'm actually not sure exactly where I'm not sure exactly where within the cells that it will deposit. Um, that's a good question. I would need to check and see.
Thank you.
It looks like Stephanie asked if there was I mentioned a handout. So Chelsea just included the flyer for anybody who was not on that email into the chat. So you guys are going to be able to access that. And then if not, she would be able to email that to you so that you guys have access to the handout. And then Nancy asked about the slides. So those will be on that same link that Chelsea included earlier in the chat. So those will get shared there as well as a recording of this session. Can you elaborate on saturated fat some more? So Helen is asking about daily cooking, what percentage of saturated fats should be used? I am not sure what the actual percentages are, so I'm not a dietitian or nutrition student, so I'm not sure exactly what those recommendations are, but I'd definitely be able to look into those and let you guys know next week. Oh, and Chelsea included the MyPlate website. So this was something that I recommended last week. So this is a really great resource for kind of putting in your information, and then it'll give you recommendations based on what the current guidelines are for certain foods.
Could you explain again the relationship between blue light and was it macular
Yeah.
carotenoids?
Yes. It looks like Chelsea's going to go back to that slide for me. Basically, so the way that these carotenoids deposit in the macula basically is for protection against oxidative damage that we get from harmful high wavelengths of light, low wavelengths of light in our daily living. And so with the deposition of these carotenoids, it basically means that blue light is going to be absorbed differently if you have higher levels of these nutrients. And so when we're looking at this in terms of like actually assessing how much of the macular pigment is in the eye, we use different wavelengths of blue light flickered back and forth between one another. And then we're looking at a ratio of at what point the participants can't see the blue light switching back and forth between the two different wavelengths. And so it has to do with how the carotenoids allow us to absorb these low wavelength lights.
When you say macular pigment, are you referring to the deposits of carotenoids?
Yes.
Okay. Thank you.
Yeah. It looks like Sarah is asking how Keto Diet fits into this information. I am not very well versed on Keto Diet. Keto Diet I know has like higher levels of fats than so it looks like Kelsey also just answered. As high levels of fats, we don't really know how much we don't really have much information on long-term effects of keto diet with relationship to cognitive function. The primary research that's been done in relation to keto diet has been primarily in people with epilepsy, where that specific diet pattern affects epileptic seizures. Looks like Jackie is asking how a teacher can help with nutrition in students. Yeah, that's a really great question. So Chelsea is including mentioning some resources to kind of teach kids a little bit more. But yeah, that's something I think people who study nutrition like really want to see more of in schools, is just proper nutrition education, so that kids are a bit more knowledgeable about what they're eating and how that can actually affect their bodies a bit better. So I think like looking into resources a bit more in nutrition education, and maybe looking to see if there are people in the community that would be willing to teach a bit more about nutrition are really great resources. What is normal water intake for kids? So I think the current recommendation for water intake is 2 liters a day. Water intake. Sorry, I'm actually just looking it up right now. So it looks like, according to the Mayo Clinic, they're recommending a bit more than 2 liters per day. Yep, and it looks like Chelsea actually just put that in the chat too. You guys have any additional questions? That's a lot of bathroom time. Yes, when the kids came into the lab, when they were on, you could always tell when they were on the high intake day, because we had to take breaks a lot for them to use the bathroom. Yeah, it looks like Robin also included some resources for children nutrition in the classroom. So that's really useful. Yeah, I think that was something that was kind of highlighted as we were doing this study, was that kids are being restricted water through the day because teachers don't want the interruption of bathroom breaks during classes. And so that's something really important, that like kids really should have access to water during school, because they're basically becoming dehydrated during school, since teachers don't want the interruption of bathroom breaks.
Are we able to get into this websites that you're providing us after we're done with this, or no?
I'm not sure if the chat will be available, so maybe Chelsea, if we could find a way to include that information on the extension website. All of my resources will be linked in the slideshow, so you guys will be able to access the papers, but maybe I can go through and just edit the slides to include these links so that you guys have access to them as well.
All right, thank you.
Are there classes for this education for school during the school day? Sorry, Jackie, what do you mean for, are you saying like classes for teachers to take to include this information? Yeah, I'm not sure. That's a good question. I know that there are classes for students on campus. So I know there's like a nutrition communication class that a lot of the nutrition students take, but I'm not sure if there are classes for the public. Oh, Sherry just included SNAP ED, so it looks like maybe there's a program through SNAP to help kind of teach more. Thank you, Sherry. Do you guys have any additional questions? Like Chelsea just linked again to the surveys as well as the recordings for this week. And this Discover Wellness series for you guys to access as well. Okay, so I think if there are no more questions, we can go ahead and end for today. It looks like Ophelia also just added Eat More Save as a website for tools to reach out for nutrition work. And Chelsea just included the actual link for extensions Eat More Save. And I'm going to be including, I'm actually editing my slides right now to include all of these links so that you guys have access to them, since the chat will probably disappear after the session is concluded. Okay. Do you guys have any additional questions? Okay, well, I hope everybody's staying healthy and doing well. And if you guys have any additional questions, go ahead and reach out to me or Chelsea, and we will definitely be able to help you guys find the answer to those questions, either at future sessions or just next time. And Chelsea, if you could leave the session open for a little, I'm just going through and adding these links.
Okay. Thank you, everybody.
Thank you.